Everybody, we're going to do a lecture on conservation for biodiversity. This is C4 from our option C that we're working on. So here's a picture of a Amazon pink river dolphin. They're beautiful. I've actually gotten to see them when I lived in Brazil, and they would swim right up to you in the river. They're beautiful animals, but they are an endangered species. So when we get to the endangered species assignment, if this is an animal you're interested in, keep it on your mind because you could choose to do that. So let's get going. So this is the IUCN Red List of Threatened Species page, and you can see here it shows you the different levels of extinction that they have. So least concerned, near threatened, vulnerable, so this species in this picture is considered vulnerable, or VU, endangered, critically endangered, extinct in the wild, or extinct. So when you hear us talk about endangered species and conservation, you'll hear a lot of different levels, and this is to let you know of what level of concern they are, and this helps people decide what types of conservation methods and level of conservation they need to do for certain species. So you'll be using this page, the IUCN Red List, when you do your endangered species brochure. But I wanted to go over the terms so that you're aware of them as we talk about this in the lecture. Okay, so just to kind of see a trend of what categories of species are struggling right now, this is the IUCN Red List Index of Species Survival. So notice it goes from a 1 down to 0. You don't see 0 on this picture. And this is going over the course of years. Oops. So what you're seeing here is a decrease in corals. So they are having an increase of extinction. Mammals, there is a decrease, but not as severe as corals, for example. Amphibians have more of a decrease than mammals, and birds are kind of similar in the sense to mammals in that they're just a consistent pathway or a consistent line, which means that, and if you see, they're pretty high up here in the species of survival level because an RLI value, which is the Red List Index, RLI, of 1.0 equates to least concern. So the higher up you are here, the least concern you are, and the lower down you go, the more of a concern you are. So notice that the line for corals went from least concern in 1997, if you come down to the years, to an extremely, their speed of their line decreasing is made them higher and higher of a concern for their survival rate. This slide here is showing the increase in the number of species that have become threatened. So this is how many the IUCN, which is the page I showed you, the IUCN is assessing this many species, and this is how many total of species over the years have become threatened. So notice that it has increased. This is a picture of the dodo bird. But what this slide is showing you is Brown University in the United States, which is an excellent university, figured out data on the rate of extinction for species. So basically, before humans existed, what it seems like is there was probably one species per million species per year that went extinct, where now what they're saying is the new estimate finds that species die off as much as 1,000 times more frequently than they used to. So that's 10 times worse than the old estimate of 100 times. So species are decreasing or becoming extinct very quickly now, and they're attaching that to human causes. So here's the dodo. The dodo is a famous example of extinction. It was one of the first ones that kind of made people realize that species can go extinct from us. So the dodo is from the island of Mauritius, which is a southeast Africa. It's on the eastern side of Madagascar. And why did it die? The arrival of the Dutch in the 1600s which led to deforestation of the island, and there was a lot of invasive species like cats and dogs and rats that often come with humans colonizing an area. The dodo are actually flightless birds. You can see their little wings here. And so they had very short legs, so they couldn't run away, and they couldn't fly, so they had no adaptation to avoid predators. So let's get into some of our understandings here. So this is understanding 6. When we talk about biodiversity, we talk about 2 different vocabulary terms, richness and evenness. So when you look at these pictures here, which one do you think is most rich? Or which one, sample A or B, do you think is most even? You can see here that here it is more rich because there are 3 different species in this picture. But it is uneven because there are more of one species than there are others. But it is still considered more rich than sample B because there's only 2 species in this picture. Sample B is more even though because you can see there are 4 of each of the species. So when we look at these types of numbers, it helps tell us something about an ecosystem. How diverse it is means it's more healthy. So the more biodiversity in an area, we would say that this ecosystem is healthy. So technically, this ecosystem is more biodiverse, so it must be healthy. But so is this one because it's even. Something's happening in that ecosystem that's supporting different species to be of equal population sizes. So high levels of both of these things, richness and evenness, represents a healthy biodiversity. And just to kind of go back to that term biodiversity, that means diversity is having differences, right? Different kinds of species of life. So life diversity. Okay, this is understanding 1, which is about indicator species and biotic indices. So an indicator species is an organism whose presence or absence or abundance reflects a specific environmental condition. So when we see these species, an indicator species, we know that something's going on in this environment. We can tell how healthy the environment is, or if it's absent, we can tell something's wrong with this environment, that it's not healthy. So lichen are extremely important to a lot of species. I think in some of your ecosystem presentations, you brought up that it's a keystone species to some, think in the tundra. And lichen are very sensitive to different nutrients in the soils as well as gases in the atmosphere. So their presence lets you know that the levels of these things, of these nutrients and gases, are healthy. When they're not present, you know that they are not at healthy levels. This is a caddisfly, or different microbes tell us when they're present in an ecosystem if it's healthy or not, because we know of their food webs and we know, or their food chains, and we know who and what species would be present if they were there. So the caddisfly, when they are present, it's usually a healthy ecosystem because different fish depend on them as an eating source, or they are there to break down, they're decomposers, they're there to break down different nutrients in the soil. So when they are present, we know that the ecosystem is cycling the way it's supposed to. So they're indicator species, both of these, to give us an idea of how that environment's condition is doing. We can use these things, or these species, to help us create a biotic index. So here's how we can calculate a biotic index, which is understanding 2. This is a picture of kick sampling here, which is basically you put a net into the water and you let the flow of the stream gently push organisms into the net. So you're not picking them up or harming them, they're flowing into the net on their own, and then you count what ends up in the net. Okay, so kind of like quadrant sampling, except you're not laying down a square and counting what's in the square. This is what you can do in a water environment. So kick sampling. So you could do this for an IA if you wanted to do it. So a biotic index is a measure of the health of an ecosystem. So you could figure out how healthy, or compare how healthy 2 different bodies of water are here in maybe botanical gardens for an IA. So a biotic index is a measure of their health. Intolerant species score highly. So a clean ecosystem will have a higher biotic index than a polluted ecosystem. So remember, intolerant means that species that can't handle a lot of pollution, they're not tolerable of it. So let's make a scale here, 10 to 1. This species here is a 10, meaning that it's intolerant of toxins, so it can only handle clean water. Where this species here is a 1, because it can tolerate very polluted water, it can live in very polluted water. So if we see this species, number 1, we can assume that that water is probably very polluted, and if we see this one, we know it's very clean. This is called the Simpson Diversity Index, which we'll use later. This helps us figure out how diverse an ecosystem is, where this one helps us figure out the biotic index. Okay, so you would do both of these if you were examining how healthy an ecosystem is. So we're going to look at the biotic index first. So notice the equation, BI stands for biotic index. You take the sum of NI, which is the number of individuals of a species, so 10 of this species, for example, or 12 of this species, times the AI, which is the tolerance rating of the species, so whatever number they are given, between 1 and 10 for how tolerable they are of clean water versus pollutants, and then you divide it by the total number of individuals collected. So let's run through an example here. Okay, so we're going to compare the biotic index of 2 sites, A, which is upstream of this village or town, and this is B site, which is downstream of this village or town. So this water is moving down through this direction. Okay, so you might assume that this water is cleaner than this water because this water has to go through this village. So here is some data that's been collected from site A and from site B. So you have to figure out the BI for each of these tables. So it's kind of like when we did the chi-square with the table here. So here's the number of species that were collected at site A. So there was 6 different species. And this is how many of each of those species were collected. So 12 of species A, 23 of species B, and so forth. The total of how many were collected represents N, which is the total number of individuals that were counted. So this would be 83. The AI is the level that you gave, or that has been, if you can research it, for how tolerable they are from clean water to polluted water. So this was something you would have to research. So species A is a 10, species B is a 9, and so forth. This is doing this part of the equation right here. So Ni times AI. So you would do it first for species A. So 12 times 10 gives you 120. Then for the next one, gives you 207. 21 times 8 gives you 168. So then you have them for each of the species. Then you need to sum them up. So you would add these all together and you get 652. So what's left is you have 652 for this top number, and then you need to divide by the total number of species collected, so that was 83. So if you do 652, so I'm here, divided by 83, that'll give me the biotic index for site A. So here you can see site A was 7.9, and site B was 3.8. Okay, so what you would then also want to look for, to go back, so the higher the number, the more healthy that site is. So in this case, we would say that the site A, upstream, was more healthy than the species that were found in site B because of what species were found in site B versus site A. So higher is better. Okay. So now we're going to look at the other calculation, which is the Simpson's reciprocal index of diversity. So the higher the number, which would be D here, would represent how diverse the ecosystem is. So we're going to compare these two pictures using these as our data to figure out which one is more healthy or which one is showing more biodiversity. Okay, so here we go. Looking at the equation, we have N, which is representing the total number of organisms of all the species. So it would be N, which is N minus 1 times N, divided by the sum of little n, which is the number of organisms of a single species, times little n minus 1. So here in this picture, we're going to compare them. Here's our data set. So we have for species A, which you can assume is the butterfly. There are 6 of them. Species B could be this dragonfly, there is 1. And then species C, the ladybug, there's 1. There's a total number of 8. This would represent N. In this sample, for sample B, you would see there's only 2 species. So we have this moth, and we have this, I think it's an ant. So there was 4 of each of them. That's 8. So let's look at sample A first. So first you're going to do the total number of organisms representing N. So you can see here the total number is 8. So 8 minus 1 is 7 times 8. That's going to give you 56. Then you have to do this for each of the species here. So remember, little n represents number of organisms of a single species. So here we have 6 of this single species. So it's going to be 6 minus 1, which is 5, times the little n again, which in this case was 6. So here we're going to have 30. So then you can see 30s here. Then you have to do the next species because it's sum of, so we have to add them all together. So here for the species B, it's 1 minus 1 times 1 is going to be 0. And then for species C is here. So 1 minus 1 times 1 is also 0. So then you're going to get 30. So 56 divided by 30 is going to give us the Simpson's reciprocal index, which is D. So that's going to be 1.87. So that's how you would figure it out for sample A. Now I'm going to do sample B. So here is sample B. You can see there's the 2 species. It was 8 total. So you would do big N here. So that's the total number. So 8 minus 1 is 7 times 8 is 56. This is a coincidence that they were the same in each picture. Okay, this could have been a different number. It does not always end up being 8. See here, in the last one was also 8. It's just a coincidence. Okay, so then we're going to do it for each of the individual species. So it was 4. 4 minus 1 is 3 times 4 is 12. So there's that. Plus the next species, B, 4 minus 1 is 3 times 4 is 12. So that's going to be 24. So 56 divided by 24, because it's the sum of these two. So 56 divided by 24 is going to give us D, which is equal to 2. So since sample B is a little higher, we would say it has a slightly higher biodiversity, which would mean that if we go back to this picture, sample B is a little bit healthier. Probably because it's more even. Remember, species evenness versus species richness. So the evenness must have made it slightly higher, but they both are healthy, technically. Okay, so what are ways that scientists use to determine the health of an ecosystem? To review, it was looking at the biotic index or calculating the Simpson biodiversity index, and looking for species richness or species evenness when they count how many species are in that area. Okay, so now let's look at understanding 5. So geographic features can affect the biodiversity of an area. Some features geographically are the size. So if it's a large size versus a small size. So think of an island, for example. The larger the island, the more resources there's going to be, the more breeding sites or the more hiding habitats they can have, the more niches they can be in. Large sites will avoid extinction with unexpected factors like fire or disease as well, because they have more places to hide. So when you think of a large island versus a small island, a large island is going to be better. Now, if we're talking like a a larger continent is going to be better than a smaller continent. So in this case, size for geographic features. Larger will help biodiversity, smaller will not. Another thing that can affect biodiversity is edge effects. An edge effect is the edge of a habitat. So it's the boundary between habitats or ecosystems. So for example, if you look at my cursor right now, this is the edge of this habitat. This habitat, this would be the edge here, and this would be another edge. So see there's an edge in the middle because this road goes through this habitat. And this would be the edge here for this habitat. So there's certain, we call it an edge effect, because certain species can handle living on the edge where others cannot, and they need to be more in the center. So they're more, this would be their optimum zone here. But others do very well on the edge. But usually edge effects decrease biodiversity. So you don't usually find as many species on the edge. And if you are finding species, they're very tolerant species. They can handle pollution. They can handle a lot of interaction with non-native species that are being introduced. So at the edge of the forest, there's more sunlight, more wind, and less moisture than the center. So usually it decreases populations of species on the edge. So they do better here because they're protected from the wind or extreme sunlights, drying out of soil. Forest fragmentation, so this would be fragmentation here, is when there's a splitting of the ecosystem. So splitting in half by roads, or maybe a farm was put in the middle of that habitat, that fragments or makes it into pieces. So now the edges have increased. You can see here, now there's an edge here and here because of this fragmentation. So that can affect, now there's more edge type species instead of the species that would be in the middle, like here. This is another example because it's an elongated shape, okay? But you can see that this, because it's elongated, it's going to have different edges here. There's not this thicker spot in the middle where you would have more species that are protected and can have an optimal zone. So an exception to this rule about edge effects affecting or decreasing biodiversity, cowbirds actually thrive at the edge because they lay their eggs there, and they do very well there, and it would increase the cowbird population. But that's an exception to the rule for biodiversity. This is going over the same thing. So here's a picture of the cowbird here. And so they're the ones who are the exception to the rule. This is showing you a natural reserve is an area that we use to protect animals, but on the edges of the reserve, you're going to have that edge effect. So maybe there's a village nearby that's going to disturb the species that live on the edge, so it pushes species further in. This would be a fragmented reserve, so maybe a road goes through it. So the extent is going to be worse for the biodiversity. It's going to decrease because now there's more edge to this reserve. This is showing you the great ape habitats. This is today, but you can see here by 2030, they believe that they're going to be very fragmented. So the green areas are representing their zones, but you can see with the red area, with farmland and things like that, it's going to fragment them, which is going to separate them, and they won't be able to reproduce as often because they can't get to each other, so it isolates them. The third feature that affects biodiversity is corridors. So what the WWF, the World Wildlife Fund, have started doing is they fundraise and they take donations to build corridors to stop isolation of species. So one example that they did this for is the panda populations, and they basically built a corridor which is planting all these trees and making it so roads can't go between these trees, which then connects populations of pandas so that they could reproduce with each other. Here, you can see they're isolated, so if they want to come out of this area of trees, they won't. to make it to other populations of pandas, which puts that population at risk because they're only going to be reproducing with the few in this area, which will decrease the strength of their genetics. So what the World Wildlife Fund did is planted trees all along this river to build corridors. So now they can get to each other. And this will help them be able to find food if they need it, reproduce with a higher population of pandas, and access to more habitats for space and living. Here's some examples, better versus worse. So here, this would be general principles for when we build nature reserves, because a lot of times they're government funded and we want to have some protected space of land. So we design the shapes of them. So when you design a shape of a nature reserve, this one's saying larger is better, smaller is worse. This is not fragmented, so this is better, where these would be worse because they're more fragmented and there's more edge effect. If they have to be fragmented, these are better because they're closer together, so the species don't have as far to go if they need to get to each other, and they can have corridors built between them better. So clustered is better than spread out in isolated reserves. This is showing a habitat corridor, which could be built very easily here between these ones. So corridors are better than isolation. So these are some rules for governments or organizations when they're building nature reserves to benefit the biodiversity in those areas or in those reserves, so that species can keep it populated and keep it very diverse. Okay, active management of endangered species. So what we've done, or what humans are doing, is we're intervening and we've realized that we're creating a problem for species and we're trying to protect them and repopulate these species. So human intervention in conservation of an area, so building reserves and keeping people out and managing how many people can go in and what types of roads can be built, all of these are important in conserving an area so a species can feel safe and that it can reproduce there. Some techniques to conserve an area are restoration, so restore the land to its natural state. So there's a lot of research that's been done on what plants are from that area, what species should be in that area, and what size they used to be. So we can collect data to see if we're getting them back to that original size of population for plants and animals. So restoration. Recovery of a threatened species. So we might have to take a species from an area and allow it to do captive breeding. That's this one down here. And then let it repopulate, and then we can reintroduce those species back into an area. There are specific guidelines for reintroducing a species because you don't want it to become dependent on humans. So for example, a lot of the caretakers will wear large costumes that look like they're that species, or puppets on their hands to feed little birds, so they think they're being fed from their mother bird. So wearing costumes and not letting them recognize humans, because we want species to be scared of humans so they can be put back into the wild and not trust us. So they're not going to fall prey to a poacher or any hunter. And legal protection against development or pollution. So governments need to make laws, and this will help protect these species or protect their environments they live in. That can help conserve a species. And then removal of an introduced species. So going to our invasive species that we had talked about before, removing these introduced or invasive species so that the native species can thrive. So last slide here. There are two types of conservation methods when we talk about how we're going to protect a species. There's in situ, which is conserving in your own habitat, so leaving that species in that habitat, or there's ex situ, which is conserving outside of their habitat, so taking them from the habitat to help them. So the advantages of in situ are species remain adapted to their habitat, animals will maintain their natural behavior patterns, and species interact with each other, which helps conserve that ecosystem. We can keep an eye on them in this type of way by doing GPS tagging. For example, with whales we've done this, or dolphins. For ex-situ, for conserving outside of their habitat, some of the advantages are some species become too rare, so it's their last chance of survival. So we take the remaining out of their habitat, and it can allow for captive breeding programs like of pandas. The destruction of their natural habitat can be necessary to remove threatened species. And then seed banks are something that you can do. So you're taking seeds from these plants to store for replanting. So these are methods that you can do to help conserve by removing or protecting or storing species outside of their own habitat for hopefully conserving them at least so they're still alive. And hopefully one day to reintroduce them into their own habitat. So this is C4, Conservation and Biodiversity. Let me know if you have any questions. Please upload your notes to ManageBack so that I can see that you did them. Thank you.